Okay, good afternoon, everyone, and welcome to the UC Ag Experts Talk on diseases of citrus. I'm Stephanie Pereira with the UC statewide IPM program, and Cheryl Reynolds is also here to run the poll questions and troubleshoot any technical problems. Please note that Cheryl and I are both working from our homes, and this may affect the quality of our audio connection. Please also note that this webinar is designed for growers and agricultural pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, are not to be followed without a clear understanding of their legal use in home environments. And now I'd like to introduce our speaker for today. Dr. Peggy Mauk is the Director of Agricultural Operations and Subtropical Horticulture Cooperative Extension Specialist in the Department of Botany and Plant Sciences at UC Riverside. Today, she will discuss diseases of citrus. And now I'd like to pass the mic over to Dr. Mauk. You can go ahead and share your slides.
Good afternoon, everyone. It's a pleasure to be here. Again, I'm Peggy Mauk. I'm Director of the Agricultural Research Station, and we have two stations here in Riverside. The topic is diseases of citrus. I just want to give you a quick overview of where I am and what we do. We have two research stations, one in Riverside, adjacent to the campus, and it's about 450 acres. And we have a second research station out in the desert, the Coachella Valley Ag Research Station, and it was established in 1991, and it's about 540 acres, of which we have about 100 acres of active research. The research station preceded the campus by 40 some years. The campus was established in 1954. And so there's a long history of only citrus research, and then it slowly evolved into many crops. On the bottom of the slide, which is also my background of my Zoom call, it's an overview, a panoramic view of our research station. And then above it is the historic building on campus and a view from I'm not sure where that view is from, but you can see you're looking across citrus trees to the research station buildings. There were 2 or 3 buildings there, and the director had a house off to the right. I am the equivalent of that director, but they didn't give me a house. But I have a great job. I'll go over a little bit of California history. Citrus. In the state of California, we have about 255 acres of citrus, of which about 103,000 acres are very high value, easy peel mandarins and lemons. In California, our citrus market is primarily a fresh market. We do have more and more juice opportunities as Florida's production is lower because of HLB, Huanglongbing, but we are primarily a fresh market state. We're also one of the most regulated states in the nation. We not only have our EPA for the US, but we have Cal EPA as well, and a whole list of environmental regulations which affects growers economically. The sum of these points means that we need high quality fruit and high yields to stay profitable. So the topic is diseases, and so I thought I'd ask you if you know what plant pathology means. And plant is phyto, and pathology is a study of disease. And disease is a continuous malfunction, and that continuous malfunction can either be biotic, which would be a pathogen, or abiotic, which could be physiological or environmental. And the foundation of plant pathology and the foundation of control is understanding that disease triangle. So you have a pathogen, you have the environment, as an example, rainfall, and the host. And you have, it's an intersection of those 3 components that can bring about disease. So if the environment is appropriate for the pathogen, and the pathogen is available and the host is susceptible, you'll have disease. And here's a little example of the disease triangle. So you have the susceptible host, the pathogen, and the environment. And when those 3 things come together, you have that triangle of disease. If you don't have the pathogen, then you have no disease. If you don't have the environment, appropriate, there's no disease. And if you have a resistant host, you also won't have disease. So another look at disease control is manipulating that disease triangle. So a susceptible host would be one that the pathogen survives in the plant and causes the expression of symptoms. So our goal is to manipulate either the host for disease control, so we want a resistant host so the pathogen can't survive in the plant, or at least a tolerant host that the pathogen can survive, but the symptoms aren't expressed strongly enough to impact yield. The other aspect is to manipulate the environment, and that would either be through cultural practices or through chemical control. There's other ways of manipulating, but those are the two that we're focused on here. So in California, there are several diseases that are endemic to our state. Root rots caused by Phytophthora, root rots, gummosis, and fruit decay caused by Phytophthora or armillaria root rot. There's several post-harvest fungal pathogens that are endemic, and then some viruses and viroids. So citrus tristeza virus, which in the early 1900s into probably 1950s was considered the most devastating pathogen in California until we had host resistance. Citrus cirrhosis virus, which is not as common because of our clean budwood program, but is still found. Citrus exocortis, cachexia, and citrus stubborn. Citrus stubborn is seen in areas. It's not as devastating as tristeza can be or exocortis cachexia. Next slide. So I'm going to start out with the fungal pathogens. Phytophthora, there's 3 species that are important in California, and they all can cause any of these 3 items. We have Phytophthora citrophthora, Phytophthora syringae, and Phytophthora parasitica, which is also Nicotiani, and I did not correct that top bullet, as it was formerly known as parasitica. And they all infect citrus roots, trunk, and fruit. Citrophthora is active year-round. Syringae is a cool season pathogen, so at least in Southern California, it's January to March, could be December, but it is definitely a temperature cool season pathogen. And Phytophthora nicotiani or parasitica is a warm temperature pathogen, and that you'll see from July to September roughly. So it's important to understand the pathogen in order to understand how you can interrupt it or control it. So in general, if you have 18 hours of wetness, 18 hours of wetness is required for the fungus to produce sporangia and release the zoospores. The zoospores are the motile phase of the pathogen. So here's a sporangium, and it's containing the zoospores, if you can see my mouse. And here's the zoospores being released, and they swim to find a susceptible host. And a susceptible host is going to be the roots. It could be splashing, and so you might have it splashed up on the trunk, or it could splash up on the fruit. So that's why you see all 3 phases. 3 hours of wetness are required for infection. So if your sprinklers are hitting the trunk and the trunk stays wet, that's really ideal for the fungus to be able to infect if the wood is a susceptible rootstock or scion, depending on where it splashes. The zoospores from the sporangia on the ground may be splashed up on the fruit or the trunk. Inoculum levels and wetness lengths are most important predictors of brown rot epidemics. So if you have a lot of root rot and you have high rainfall for a period of time, you're set up for brown rot, which is the fruit phase of the disease. And Phytophthora populations in the root zone range from 15 to 20 propagules per gram, and that's a level where you warrant treatment. Now, Phytophthora brown rot, root rot, brown rot, and gummosis. The Phytophthora, as I said, it produces zoospores that swim in the water, and this picture is a really great example. When I first came to the research station here, I I just saw this picture perfect row of lemons. And so the trees had been planted in the middle of a furrow. It was furrowed, and then these are furrow irrigated. Sorry, my mouse is doing wrong things. And so the trees are planted here, and generally you start watering here. Well, when a tree is small, you have the furrow run closer to the trunk. And in this case, in this field, the trees were planted a little bit deep, or if they weren't deep, they settled deeply. So the soil was up on the scion wood, and the furrows hadn't been cut away from the trunk. So water was flowing essentially down the middle of the row. And so this is a perfect example of how the zoospores move. So you have a tree that's infected here, and the water's flowing, and so you see a series of trees that are infected with Phytophthora. There are several rows like this, and some of which we were able to save with fungicides, but many of these trees we lost. But I really was grateful for this because it's a really nice example to show people what happens when you have a combination of factors that create that disease triangle. So you had ample water, you had endemic fungus, it is everywhere, and you have a host that was buried too deeply, so the susceptible wood was exposed. Here's on the left of the slide is a gumming on the tree trunk, and then this is a brown rot phase on the lemon fruit. So you see it's kind of washed out looking, bleached, and that's Phytophthora is actively infecting it, and many of them are dropping to the ground. So in control of gummosis, the plant tree, you need to plant the tree so the graft union is above the soil line. And you have to assume that the trees are going to settle, because especially if you're a commercial grower, you're augering the holes, and the auger may be a little bit deep, and you don't pack enough soil below the root ball when you're planting it. So you have to make that assumption that it's going to settle and adjust accordingly. In this case, you can't see where the roots are. This is all scion wood, and you can see the gumming on the tree. You want to adjust the irrigation so the trunks stay dry. And one thing to look for if you have gummosis is when the trunk is oozing, that means the fungus is active and it's time to treat with a fungicide. So examples of fungicides that are effective is Aronis, Ridamil Gold, Alliet, and phosphonates should be added to that list. If you have more than 50% of the trunk girdled from the gummosis, it's time to, it's really makes more economical sense to replace the tree, because by the time a new tree is up and producing, it's a shorter amount of time of lost yield as compared to a slowly declining and dying tree. So I always recommend that even if you really like that tree, if you have 50% of that wood that's girdled, it's a good idea just to plant a new tree. And if you plant a new tree, you need to actively treat with fungicides to prevent new infections. So again, proper planting, you can see where the bench is on the rootstock scion here on this navel orange, or actually that's a mandarin, and you don't see the graft union on the other tree. So root rot, one of the telltale signs of root rot is you take a shovel full of soil. If you can't find roots, that's not a good sign, because either they're not watering enough to have roots, or you're overwatering and the roots are decaying very fast. If you take a shovel full of soil and when the roots are removed, you'll see the central steel of the root here. The exocortis is sloughed off, and that's because it's rotted. So that's, again, not a good sign of root health. And it's not necessarily Phytophthora, but it is an indicator that it could be Phytophthora. You want to use planting strategies to reduce a root rot. So this is a research project where we're looking for increasing production more quickly. And so we're comparing planting strategies. And so it's a really nice example of planting on the flat. You get pooling of the water, and so you get more water logging. You plant on the berm and you have healthier trees, you have better water drainage. So that's just a comparison of planting strategies on the flat versus on berms. Berms will drain more quickly. So if you know you have a root rot situation, planting on berms is to your advantage. It's not as fun to walk the field, and it's certainly more work to plant the trees, but is definitely worthwhile. In managing root rot, the first line of attack is always to go for resistance. So you want to look for resistant rootstocks: trifoliate orange, swingle, tremolo, citrange, alamo, also known as macrophylla, and sour orange are all good examples of resistant rootstocks. Fungicides that are effective, Orandis and Presidio are new chemistries, Ridamil Gold, and phosphonates like phosphite. You want to treat for the first 3 or 4 years to prevent and control root rot. If you're going into a situation where you know you have root rot, you want to treat twice a year when the roots are flushing. So that's generally June, August, September in Southern California, and you're going to do that for 3 or 4 years so that that tree has a good start. That root system has time to grow out from the root ball, from the nursery setting, and then that resistant rootstock has a better chance of surviving in the face of root rot if you've given it a good foundation to begin with. And also you want to manage your irrigation. So you want to avoid excessive wetting or excessive wetting followed by excessive drying. That alternating of over dried followed by over wet. The soil is all hard when it's excessively dry, and it starts to crack. And when your soil's cracking apart, your roots are injured. You add water. Now you have the zoospores being released, and you have roots that have been damaged, and so they're releasing root exudates, and it's very attractive to the zoospores. So more even watering is far better, and managing your watering is critical in root rot control. So management of brown rot is really prevention. So you want to prevent root rot, which is where the fungus is coming from to be splashed up into the fruit. And if you have root rot and it's not controlled, which is not uncommon. So if you have root rot, you want to make sure the skirts are pruned to 2 feet or more above the ground. This picture is an example of what not to do. And this tree, when we get rains, you'll have a lot of brown rot in there. It's a research site, and so we actually don't want to control the Phytophthora in this case because we have research studies in there. But the skirts are down at the ground, which is not what you want. If you want to control it, you want to spray fungicide between October and December, before or just after the first rain, because high rainfall equals high disease. And you want to repeat the spray in January or February or whenever there's excessive rainfall. Spray the skirts to about 4 feet above the ground. And it seems like a long way for the zoospores to splash, but I've seen fruit easily 4 feet up the tree that have brown rot. So you need to spray all the way at least 4 feet up with fungicides. So in terms of controlling, spraying the ground beneath the trees suppresses the fungus, so you have less inoculum. Copper, Orandis, Rivus, phosphonates, those are all foliar or fruit treatments. And so you can spray the trees. Later in this presentation, I'll talk about the exports to China, which there's a specific protocol that has to be followed. And copper is one of those fungicides that can be used, but you have to prevent brown rot. Orandis, Presidio, Ridamil Gold, and phosphonates are applied as soil treatments for brown rot control. And for soil applications, you can inject it into the irrigation system. So viruses and viroids. Citrus tristeza virus, we have tolerant rootstocks, and we have the Clean Budwood program, and that's called the CCPP. It's a Citrus Clonal Protection Program, and it's a state-of-the-art example for the world of how you run a citrus nursery program, is to require that nurseries have clean budwood. Prevention. So all of the most common viruses and viroids, historically growers would see a tree they liked, they would take a bud and plant it out. And if you have cirrhosis, you're planting it out and you may not see symptoms right away. If you have tristeza virus, well, that can be much more catastrophic. There's different forms of CTV. There's stem pitting, which is more catastrophic and will cause sudden death of the tree. We also have citrus tristeza virus, which is a very mild form, and that is fairly common in California, certainly in Southern California. And with the resistant rootstocks, you don't have issues with tristeza virus. But having clean budwood is of critical importance. Cirrhosis, you see cirrhosis from time to time. Occasionally you'll see it coming out of nursery grown trees. It's rare. It's much more common on trees that are, especially trees that are planted before the 60s, before we had the Clean Budwood program. At the UCR campus, we have a block of trees that's part of the original purchase of the property. So the property was purchased around 1917. So the trees are well over 100 years old, well over, because the aerial pictures look like the trees were probably 15 years old at the time. So this picture in the middle is one of our trees, and that is a beautiful example of cirrhosis. The bark cups and chips off, and the tree slowly declines. It is a slow decline. Cirrhosis, I've seen growers try to keep things economical. So plant a new tree next to an old tree, or not remove the stump of an old tree. So they're doing as much as they can to keep things going. And they plant next to a tree that was infected with cirrhosis, and the trunk wasn't, the roots were still alive. And that poor little baby tree, oh my gosh, I've never seen a young plant totally engulfed with cirrhosis. And that was a really great example of root-to-root grafting. Exocortis is also something that's not, I don't see it in growers' fields. We do have Exocortis in our 100-plus-year-old block of trees, and we keep them there because we're a teaching institution, and this is a great opportunity for people to see diseases. There's some famous trees with famous labels on them indicating the first tree that this was discovered in. So we're maintaining those as best we can. And citrus exocortis is there as well. It is more important to not have exocortis trees because it is mechanically transmitted. You can take pruning shears from one tree to the next and move the virus or the viroid. Citrus stubborn is a bacterium, Spiroplasma citri, and it is it's common. You see it more in the desert on grapefruits. We have some here. I know there's citrus stubborn up in the Lynn Cove Research Station. It causes the biggest impact is that the fruit are misshapen, as you can see in the upper left-hand corner. That is certainly impacts your pack out. The unusual thing about citrus stubborn is from year to year, it's not always evident. So the titer varies, the symptoms vary. There's years that we know the trees are infected and we can't detect it with PCR. So it's quite variable. It's certainly not the most important disease in California, but it is common. So now certainly CTV was the biggest threat to California citrus until we found resistant rootstocks. And Huanglongbing is even a greater threat at this point, and we don't have resistant rootstocks or scions. So I'm going to give a little bit of information about the disease. It's worldwide. There's a few locations in the world where it doesn't exist. Central Valley is one of those places, and we want to do everything we can to keep it out of the Central Valley. It's transmitted by a psyllid. It's a phloem. There's a bacterium that lives in the phloem, or the phloem-associated cells. It causes a slow decline, early fruit drop. There'll be some leaf symptoms, and the fruit will be misshapen and also not colored normally. Those are some of the symptoms. This picture here is from Florida. Florida it's been devastated by HLB, or Huanglongbing, also citrus greening. Huanglongbing is Chinese for yellow dragon, and you can see a yellow dragon pattern in some of the infected trees. And that was the first description, and when you're naming diseases, you go back to the earliest description. So in the US and many places they called it citrus greening, but the first published name was Huanglongbing, and so that's the official disease name. It's transmitted naturally through Asian Citrus Psyllid, ACP, and you can see different phases. You have the adult of the adult here. This is a mating pair of adults. We have the nymphs, different instars of the nymphs, and you'll see if I were to zoom in on this, which it's not a high enough quality photo, but you'll see the eyes are brilliant red after the second or third instar. You'll see this curly Q exudate, which is essentially the honeydew, and the ants do like that. And so they will mine the psyllid nymphs. And then this little bend in the leaf, that's an example of adult feeding injury. So if you see bends in the leaves like that, you need to be scouting for ACP. So there's also transmission that occurs artificially, and that's how we've gotten HLB in the United States and in California. It's through human movement of plants. So we have people that smuggle budwood in. Several years ago, I remember a story of up in San Francisco. Someone was bringing in a cigar box, and they opened up the box at the where you're going through customs, and it was bud sticks that were infected with bacterial canker of citrus. And so they could see the canker all over the twigs, and of course they intercepted that. But those things happen, and people take those plants because they're things that they wanted from where they used to live, and they don't mean to be harmful, but they end up bringing diseases to the US. So you have smuggling trees, you'll have inadvertent grafting because you didn't know you had the pathogen and you aren't checking your clean budwood sources. And so you inadvertently start moving the pathogen, or smuggling fruit that have psyllids that are inoculated with the bacterium. So citrus, the reason they say greening is you see here in the middle, the leaves are a weird greening. You'll end up with a weird greening symptom. This is zinc deficiency, and the first leaf is a healthy leaf. So you'll have a blotchy mottle, and also if you turn the leaf over, sometimes you'll have a corking, raised veins on the underside of the leaves. So in addition to the mottling, you'll have the veins thickening, or you can have the veins thickening. It's not always a sign. And also that is a sign of other viruses. So HLB disease, it prevents the fruit from developing proper color, and that's really the reason they called it citrus greening, because the fruit won't green. They'll also be half orange and half green. And so I've had people ask me, they'll say, you know what, so-and-so has a tree, and it's just really odd that the top side of the fruit is orange and the bottom side is green. And you want to check that tree for HLB, and then follow through with CDFA checking the tree and having tree removal, because that's live inoculum sitting out there. And even more devastating, HLB causes a fruit to be small, oddly shaped, similar to Citrus stubborn. The seeds will abort, and the juice is bitter. And in Florida, bitter juice can be accommodated by adding debittering agents. So you can have, while you're still having yield off those declining trees, those trees are producing fruit that have a bitter juice. Well, they debitter the juice so they can continue to have a crop and sell it. So a lot of your Florida orange juice will be debittered, where your California orange juice won't. They've done side-by-side tastings at some meetings, and they do taste similar. Of course, fresh off the tree is always superior, so it's hard to do a side-by-side in that case. The fruit is small, it grows crooked, and often uneven segments, and the seeds can be aborted. Now, the complications with HLB is it's hard to detect. The symptoms resemble mineral deficiencies, toxicities, or other diseases. You'll have visual symptoms that they take years to develop. So you may have a tree that's infected, but it may be 4 or 5 years before you see the symptoms. And you don't see the symptoms until you lose about 30% of the root mass, which, because the bacterium is actually concentrated in the roots. And so you'll have a degradation of the root system. The pathogen isn't distributed evenly in the tree, and so you have to find the right leaf to sample to detect it. The tree may be, half the tree may be completely healthy and normal, and that's the side that you sampled, or it may just be one branch or a few leaves. In Florida, you have inundative, you have a constant supply of psyllids, and those psyllids can be hot. So one tree could be inoculated many, many times every year. In California, the disease is less common. It's uncommon, I would like to say. And the psyllids aren't common in areas where you're controlling the psyllid. And so you're not getting inoculation throughout the tree. So you have to find that one leaf to know that you have the pathogen. It's challenging to collect a good sample because of that, it's hard to collect the right sample because you have to sample the right leaf. Detection and diagnosis. If you see a tree that has visual symptoms, and I know the first tree in the Hacienda Heights here in Southern California, the person from CDFA that responded to it said she knows the disease. She said from the street she knew it had HLB. It was that strong. And it was a tree that had been budded with like 20 different bud sticks that they had brought in from elsewhere outside of California. Laboratory results, so visual symptoms are important if you do see them, but there's a lag in seeing symptoms. The analysis is done via PCR. It's molecular based. It detects the pathogen. That's the only official method of detection. Research on early detection, pre-symptomatic and asymptomatic tissue, we're doing different tests. We're looking at metabolomics, which are compounds that are produced by the pathogen that are common in the tree, detecting small RNAs, ELISA for structural proteins of the bacterium, and also canine detection. So early detection is what we need. We need to eliminate trees before they're symptomatic and before they become a source of spread. HLB, right now it's not controlled because we don't have host resistance. It's not been developed into commercial varieties. We have leads on resistance, but we don't have commercial varieties with resistance. The tree removal is dependent on detection. If you can't find it, you can't remove the tree. And if it's been infected for a while, then psyllids have been on there and moved the pathogen ahead of detection. The psyllids spread the pathogen. There's an extended period of incubation when the pathogen isn't detected. So finding that right leaf is important, but there's a latency before or a very slow progression of the pathogen in the host. So it may not be detected. And so you have a host that is providing plenty of inoculum for psyllids. With the lack of symptoms until a tree is fully infected, it becomes a reservoir for inoculum. Kind of being redundant here, sorry. Right now, the canine team that was in Florida, they have 6 canines here in California that are trained to detect HLB. And it's a company called F1K9, and they're in Ventura right now. That's their home base, is in Ventura, and they can be contracted. We have a grant with the Citrus Research Board as well as USDA funding where we're evaluating the canines for detection and looking at different scenarios to help in the training of the canines. And what we have found, which is true in Florida where they've used the canines as well, is that we've taken fields that are inoculated with Phytophthora, and there's no correlation of the canines alerting on trees that are infected with Phytophthora. With or without Phytophthora, they will alert. We have a field that we've deliberately inoculated and put different fungicide treatments, and they have yet to alert in that field. So there's no correlation. We have rows of citrus stubborn trees, and they have not alerted on the citrus stubborn trees. Viruses and viroids, there's no correlation. One person asked, or many people, including myself, is, well, maybe they're sitting where another dog already sat, because you often use 2 dogs, one to detect and one to confirm. Well, maybe the confirming is because he senses the first dog sat there. So we've done different combinations, and there's no correlation. We've used female dogs followed by male dogs and vice versa. There's no correlation. They're focused on the HLB scent, not each other or human activities. We've even had them flag the trees from the other side or only GPS the trees so there's no visual. And the canines don't alert unless they're totally focused on that scent. Nobody else matters. There's to look at, we've been scanning trees here at UCR for HLB use as part of this project, and I just want to show you an example. So in March of 2019, we only ran the perimeter of this grove, and you can see this is on the southern edge of the research station. There's homes here. And we had 3 trees on the southern edge and 1 on the northern edge. 1 year later, we did only the perimeter again, and now we have 7 trees on the southern edge and 5 trees on the northern edge. So there's definitely, there was a concentration coming from the urban area. So in October, and you might need to move the box of us so you can see it, but you can see we did tree by tree survey in October just 2 months ago, and there's a progression from the edge of the south edge toward the middle of the grove, which is fairly clean, and then the edge coming from the road on the north side. And this is consistent with the research on how the psyllids move in orchards. You have an edge effect, and then they slowly start moving interior. We do have very good psyllid control. It's hard to find psyllids out there. And so I know that we're doing everything we can to prevent psyllids from coming in. Homeowners may or may not be controlling the psyllids. And so I've been encouraging CDFA to be surveying trees to the south of us, because there has to be a source. This pattern is repeated along the whole southern border of our research station. So this is being done up in Ventura. The Santa Paula Pest Control Group has hired the F1K9, and they've been surveying the groves up in the Ventura area. And they've found some very striking results. Areas that are very isolated have no alerts. Areas that are next to areas where the psyllids haven't been controlled are higher. They have growers that have gone in and removed every tree that the dogs alerted on, and the next year there was 1 alert instead of 15 alerts. In one case, they didn't remove the trees, they just pruned them, stumped them, or caged them. And the next survey, there was an increasing number of alerts. So by removing the trees, they've stayed ahead of the problem. And that's bold, because you're putting your trust in the canines to detect. And if you're not a believer in the canines detecting, you're not going to be inclined to remove your trees. But these guys are showing it works. In terms of management, the only management right now is through insect control. You want to be scouting at least 4 times a year for ACP, and you want to be controlling. You obviously want to remove infected trees, and nurseries are now required to be under screen so that there's no possibility of moving infected trees from nurseries. If there's been HLB in the area, nurseries aren't allowed to sell trees regardless of where they came from. So in Riverside, we have HLB. Home Depot can't carry citrus, or Lowe's, or nurseries. I don't mean to pick on one company. The other option is bactericides, and there is evidence of translaminar movement of the antibacterial product in the leaf. If it's applied to the leaf when the psyllids are active, they ingest the bactericide, which would kill the bacterium in the psyllid, and it also affects the health of this and longevity of that psyllid. That's been shown in California as well as in Florida, that the psyllids don't survive without that symbiote, which is really good for growers and for citrus. When the risk is high, you could use a bactericide applied as a preventative on the perimeter of the grove. There's a lot of talk about bactericides and antibiotic resistance in humans, and the studies for 7 years now, where they're repeatedly applying the bactericides, there's no resistance developing in the bacteria, the resident bacteria in the trees and on the ground. So they're looking at the microbiota of the leaf and anything on the soil, and they're not seeing any change in resistance. It's also no residues at harvest because of the 21-day pre-harvest interval, but the bactericide is broken down with UV light, and so there's no residues on the fruit. So the resistance movement from citrus to humans and animals isn't a factor. It's more of a political issue or a personal decision that you don't want to apply bactericides, but they are effective in controlling the pathogen, especially in California, where you're not getting inundative inoculations. In Florida, you're getting inundative release inoculations with the hot psyllids, and so a bactericide has a chance, but it's not going to have as much success as in California, where it's an occasional infection. If you had a lot of vector and a lot of disease pressure, you would want to treat the entire orchard. If you have only weak vector ingression from sides, you might want to just do the border. Or like in Riverside, we tend to have a problem on the southern edge of the research station, and that's also the prevailing winds, maybe a border effect down there. You have to rotate it with different chemistries, or you're going to get resistance to the bactericides and the pathogen. And there's you really need 6 applications, 4 in the spring and 2 in the fall. And you have to read the label as to the timing. But of course, it is a 21-day pre-harvest interval. Transitioning to political diseases, they are diseases, and they are political because we use diseases and pathogens and insects as part of bargaining with other countries. And that may not be the case here, but it often is the case. And so Septoria is an issue for exports to Korea, and Phytophthora brown rot is an issue for export to China. And if you want to export fruit to those markets which are lucrative, you need to be following the protocol and the program so that your fruit is approved to go to those markets. Septoria leaf spot. Septoria causes a leaf spot as well as fruit spots. It's a disease of leaves, twigs, on oranges, on fruit, on oranges, lemons, grapefruit. It occurs in many growing areas, many countries, and it's almost always associated with cold injury. So you may have ice injury on a fruit, and the Septoria will come into those injured areas. And you can see early symptoms on the top and advanced symptoms on the bottom fruit. Now, you have to have an integrated Septoria management. The pre-harvest applications of fungicides, which are copper products, and as well as you can use new fungicides like Quadris Top, Preaxor, Luna Sensation, or PhD. Those are all pre-harvest applications. You need to do this before you have accumulation of temperatures below -1 centigrade. It also forecasts based on total rainfall. So you have to scout and sample for the disease, and you submit your fruit samples to the NAVEC detection lab for molecular detection, and that's at the Kearney Ag Center. If you're planning to export citrus to China, you must follow the GAP procedures, and this is Phytophthora management. I'd already talked about Phytophthora brown rot, but I wanted to connect this to the more political or export pathogens that are issues. So you need to follow the Phytophthora management program, and there's protocols available online. I'll give you the link to the agency that will help you with this. But there, you have to control, you have to pre-treat before rainfall, you have to follow so many days after so much rainfall, you have to reapply, and you have to be able to document that you have been following this protocol. There's a Phytophthora monitoring record that shows you what you have to do. You have to make sure your skirts are pruned to 2 feet above the ground, that you've sprayed 4 feet above the ground. You have to, follow these checklists. So if you're planning on exporting fruit to China or Korea, you need to contact the California Citrus Quality Council, CCQC. And Jim Cranny is the president. Carlene Price is executive assistant. She is the one that will send out meeting notices, and so you need to attend a meeting in your area. They'll have meetings, and this year they're all remote, so it's even easier logistically. But in a normal year, you would have a meeting in Ventura, probably in Ventura, Tulare, Riverside, etc. And the website is there, www.ccqc.org. And so I encourage you to contact them if you are planning on exporting fruit. It's a good market. It's a strong market. The returns are very good in most years, but you have to make sure that your fruit will qualify for those exports. Dry root rot, which is presumed to be from injury from gophers. That's what I've always heard. There's nothing you can do to cure it. Control the gophers is a good thing to do. Some people say that injury to the trunk from weed whacking, but it's a very sudden decline. You can see the leaves are dried on the tree. It's not like they've slowly dropped off. The green leaves are dead on the tree. So it happens very quickly, and if exposed wood, you'll see a purplish brownish color. Alternaria rot and split navels. This is a big problem when you have rainfall and your fruit are enlarging. You have cracks in the navel end, and the alternaria will get inside. It's a very stress pathogen, and extreme dry, like the hot, dry winds that we get in the fall, can dehydrate the fruit, and then when you water, they swell and the navel end can crack open, and it's a perfect opportunity for alternaria to get in there. There's no chemical control, and sometimes it's not detected until it's gone through the packing house. Penicillium blue mold and green mold is very common, and that slide just makes me want to sneeze. You never take fruit off the ground in an orchard, and you need to watch your pickers that they don't, because one infected fruit in a bin, it was going to nest into the other fruit, and you're going to have a mess. So you minimize injury on the fruit. You don't put injured fruit in bins. You don't take fruit off the ground. Early infections are impossible to decline, so it comes to post-harvest control and the pathogens in the packing houses is the best line of attack, but keeping the infected fruit or injured fruit out of the bins is critical. And that's it.
Okay. So we have a lot of questions, so I don't think we can take any more. We have about 20 right now, so we'll try and roll through them pretty quickly. There's one question that asks, I've read that oxygen puts downward pressure on Phytophthora. Is this true? And does oxygenating water or any other techniques help as an environmental approach?
I think not. I haven't seen any literature that would say that. If you have anorexic soils because of excess water, the roots will be decaying because of the anorexia, not because of Phytophthora. I don't see an association of Phytophthora when you have oversaturated soils, which would be low oxygen. So I think that oxygen is not a bad thing. You know, if you had more oxygen, you'd have more pore space. So you'd have better drainage. Oxygenating the water, I don't think that would be an approach for control.
Okay. And then we have one question that asks, resistance to phosphorus acid, can a paper be supplied on that? And I think this is an extension of the question, is that different from phosphonates?
Yeah, phosphorus acid is a fertilizer. It's a very effective fertilizer, and it doesn't have any effect on Phytophthora. It's the phosphites that are effective. And so the phosphonates or the phosphites, I see it written down in different places, and it's a phosphite that there is evidence of resistance. I don't know how widespread the resistance is, and I'm sure I can find a paper that would tell you that. But the phosphorus acid is a fertilizer, and that's something that you should use.
Okay. Christina asked, when applying fungicide, do you apply to the trunk as well as the soil around the trunk, extending to the edge of the canopy?
Uh, if you're if you have gummosis, yes, you need to be applying it to the trunk and the soil surface. If you have brown rot, you're going to want to put it on the trunk just because you're going to be spraying the skirts. And so it's going to be on the trunk in that case. If you only have root rot and you apply it through the irrigation system, it's going to be on the soil surface. So the critical part of applying it to the trunk is if the trunk is, if your emitters are hitting the trunk and you're seeing gummosis, then you have the potential of the fungus infecting. It doesn't, if you're doing spraying the trees, you're going to be hitting the trunk. If you're spraying the soil surface, you're probably going to be hitting the trunk. And if you're injecting it through the irrigation system, any water hitting the trunk is going to apply it to the trunk. So I guess the answer is yes, you can apply it to the trunk. It's not going to be an added time because you're already there, and it's not going to hurt.
Okay, and we have a quick question here. Does nymph exude contain Liberibacter?
No, the exudate is just a sugary substance from the tree. I don't believe there's the bacterium is alive in that exudate. It's living in the gut of the insect, and so it's being injected into the tree with through the mouth parts of the insect.
Thank you. One person asked, where could samples be sent for HLB testing?
You should call your local ag commissioner or CDFA. There is saveourcitrus.org. There should be a link on that for where you could submit samples, but it's through the state that the testing would occur. I see the question on, do young leaves often uncurl? If the curl or the bend is permanent, it's not going to outgrow it. That's not an issue. It just is an indicator that something's been feeding on that leaf.
Great. There's one question that asks, are Phytophthora and HLB synergistic?
Yes, there is literature out of Florida that says root health is critical in keeping the trees free of HLB. I don't think free is the right word, but a compromised root system is a weak tree, and a weak tree is going to be more prone to infection. So yes, some people believe controlling root rot is a first step in controlling HLB, and I think that's true because a healthy tree is going to be is what you want to begin with, is a healthy tree. Also, a well-fertilized tree is a healthy tree, so you want to do everything you can to keep the trees strong.
Okay. There's one person who asked, I'm in an HLB non-quarantine zone. I have had ACP on my trees and they were treated. Should I assume that they are infected?
No, you shouldn't assume they're infected. What do you mean a non-quarantine zone? So that means you don't have HLB in the area. If you could type what area of the state you're in, that would be helpful. But most likely, you need to control the psyllids. And if you have questions, you can ask CDFA to sample and make sure your trees are clean, or hire the canines to survey them.
Okay. Christina asked, you alluded to citrus stubborn as producing fruit that is misshapen or deformed. Is this correct?
Yes, that can be a symptom of citrus stubborn. It's also a symptom of HLB. They're a little bit different though. You don't see, it's different magnitudes of misshapen. But yes, lopsided fruit are common with stubborn.
One person asked, is the use of translaminar carrier adjuvant recommended for translaminar fungicide movement, such as pentrabark?
Not familiar with pentrabark, and many fungicides are recommended to use specific carriers or adjuvants. If you're doing bactericides, there's a specific adjuvant that's required because it increases the translaminar movement and it increases the efficacy. But you have to read the label. It will say either to use an anionic or non-ionic surfactant, and that would be to aid in the either the efficacy of the fungicide or the spreading of the fungicide on the leaf or movement within the leaf. I don't know pentrabark though.
Okay, thank you. Marcos asked, are there preventive sprays to inoculate the tree from HLB?
The only controlling the psyllids, and the only other preventative would be to do the rotation of the bactericides. There's no other options in terms of chemicals other than keeping your tree healthy.
Terry asks, is there research on using endophytic species of bacteria similar to Paraburkholdia phytophthorans for Pierce's disease in grapes?
There is research at UCR by 2 or 3 different researchers looking at endophytes for controlling HLB. It's early on in the research, so they don't have results, but they're taking the same approach, hoping that they can control it with endophytes.
Okay. When are the peak times for ACP?
There's several. June is when I start to see them more commonly, but the spring there'll be a flight in the the slide said to check 4 times a year, and I would say that's a good idea. Depending on if you have a warm winter, you might have psyllids around in February, January, February. Certainly in the spring when you have a flush, because the psyllids are going to lay their eggs on the new flush. So anytime you have a new flush, you're going to want to be checking for psyllids. So spring, early summer, you'll have a flush, or midsummer, and in the fall. The area-wide control is one of the key times for area-wide control is during the dormancy, because you're going to be hitting the adults as a rest for the winter. And so we tend to do that in January, and we try to get all the growers in the area to treat, as well as the state to treat our area, our adjacent homeowner trees at the same time, so we have area-wide control. But that would be a winter dormancy spray for the adults.
Okay. One person asked, how many false positives do they get from the dogs? I assume not all dogs are as equally effective.
They're showing 95% effective. And so there are confounding factors in the environment. So if you have a really hot tree and a breeze that's moving the scent, there'll be a mix-up of which tree that the dog is alerting on, and we've seen examples of that. And so we come back at a different time of the day when there is no wind, and you can triangulate to where that tree is. So that would go into the false positives, because the one tree was alerted on by one dog one day and not the next because of the shifting of the wind, but also planting density is an effect. So 95% effective in the field in California is the data that I've seen.
Great. We just have 3 more questions. We've gotten through these really quickly. There's one that asks, what causes quick decline?
That is citrus tristeza virus. And I should have put that on there, on that slide.
All right, thank you. I'm not sure what this question is. It asks, it has ... about the curled leaves on new growth.
I think
How
it
does that
relates to the previous question on the, I'm assuming it has to do with the new leaves and if the curled leaves will stay curled. Now you'll have curled leaves on new leaves from the citrus leaf miner. It can be from the psyllid feeding. You can get distorted leaves from thrips. So, and those are permanent conditions. I
Okay.
don't, even aphids can cause some distortion. Is there any promising cure for HLV in the near future? I think there is promise, because we are finding hosts that are resistant. They're just not they're just not, it's the breeding that slows it down. And if we could find the exact gene and do gene editing, it would speed it up. But we need to find the gene for resistance. But Australian finger lime is very tolerant, and Citrus glauca is tolerant. And so the breeding efforts are so focused on those two citrus relatives. But we're not into commercial varieties yet. We're getting there. There is hope. We are also different than Florida. We don't have the constant inundation of psyllids. We don't have as much inoculum. And the disease appears to take a longer progression for decline. So that in itself is a lot of hope.
And then I think that is all. Thank you, Peggy, so much for presenting today. And thank you, everyone, for attending.
Thank you.
Have a great
It's
day.
been fun.